Hey everyone, we're going to take a look at the second part of photosynthesis, the Calvin cycle. And if you're wondering, there's Calvin. All right, a little review of the light reactions. Remember, the whole idea of the light reactions was to get ready to make sugar. So the light reactions was all about generating ATP and generating NADPHs because we want those H's, which have the hydrogen proton and the electrons. And that is what we call reducing power. Remember, when something gains an electron or gains a hydrogen, it's called reduced. So that's what we mean by reducing power. Basically, we're going to be giving something potential energy that way. It's like money in the bank, money in a piggy bank. And now that we have ATP and NADPH, we're going to be able to build stuff. And the stuff that we're going to build is sugar through the process of photosynthesis. So the beginning of it was photo, light in the light reactions being used to generate ATP and NADPH. Now we're going to go to the synthesis part where we're going to make sugar. We want to make glucose. Remember, glucose is a carbohydrate. It has carbon, hydrogen, and oxygen. So what materials are available for us to do this? Well, carbon dioxide, and carbon dioxide is in the atmosphere, which contains a carbon and an oxygen. So what we're going to need to do is add H's because we want to make CO2 into C6H12O6. That's called carbon fixation. When you take carbon out of the air, out of the atmosphere, and you join it to an organic molecule like sugar, that's called fixation. You're fixing carbon. And we're going to use NADPH. That's where we're going to get the H's from. We generated those in the light reaction, so we're going to take the H's from those and put them onto carbon dioxide to make glucose or sugar. So that will generate then some NADPs, meaning that can go back to the light reactions to get regenerated to grab more H's so we can keep this cycle going. So I like that diagram right there. So basically, we're doing carbon fixation here, and we are reducing CO2. Whenever you add an H or an electron to something, you reduce it. So we are adding H's to CO2. We're reducing CO2. Here we go. So CO2 by itself has very little chemical energy. But if we were to add H's to it, like C6H12O6, that has a lot of chemical energy, a lot of potential energy, and it's because of those hydrogens. We know how valuable those hydrogens are with their proton and electron. You can do a lot of work with protons and electrons, right? Because protons can be used to run through ATP synthase to generate ATP, and electrons are really good at running electron transport chains. And so C6H12O6 with all those H's in there has a lot of potential chemical energy to it. So this synthesis that we're doing here in photosynthesis is an endergonic process, meaning since we're putting the H's into it, we got to put energy into it. And so this is like uphill steps in order to get this accomplished. Each are going to be catalyzed by, of course, specific enzymes like we're used to. Everything's done by enzymes in the chemical world. And of course, we're going to use ATP and NADPH. All right, so now we're going to move to the stroma. In the light reactions, we were in the thylakoids. Now we move to the stroma. And our ATP and our NADPH will go there as well. And so this is maybe the most valuable diagram in the whole set of notes here on the Calvin cycle, because it shows us the Calvin cycle, it shows us what's happening, and it kind of keeps track of our carbons. So that's what these little circles are with the C's. We're keeping track of carbons. And I'll get them all on here to begin with so we can take a look at what's happening. I got to get myself out of the way. And so here is the whole process of what's going on. It looks kind of complex, but it's actually not too bad once you get down to it. All right, and so I'm going to go back now and let's take it step by step. So we have a cycle of molecular reactions happening. So that means we have a molecule coming off of the cycle. And that molecule is a 5-carbon molecule called ribulose bisphosphate. That's kind of a long name. So usually we just say RUBP, a 5-carbon molecule coming off. And then we have carbon dioxide in the air, in the atmosphere. That's a 1-carbon molecule. And we want to combine them. We want to fix that carbon from the air onto RUBP so then we can take a turn of the cycle. And so maybe, I don't know, this might be the most important enzyme in the world, possibly. It's called ribulose bisphosphate carboxylase. I think I just spit there. I got so excited. Or Rubisco for short. Such an important enzyme. It's the enzyme that actually grabs carbon dioxide out of the air and grabs RUBP, puts them together, like remember a pen with a cap. One of them's carbon dioxide, the other one's RUBP. So Rubisco would be me, and put them together. Now we can go about our cycle. All right, we're keeping track of carbons. We actually have 3 molecules going because it takes 3 turns of the cycle for what we're doing. So that's why we've got RUBP written 3 times. We've got carbon dioxide, we've got 3 of those. And then after we join them together, we have 3 of these. Each of these are a 6-carbon molecule. What's their name? Who cares? I don't want to know their name. So also keep in mind this is in color so we can see and keep track of where these carbons came from. So any of the red ones are the ones, at least from this turn, that have directly come from the atmosphere. So these 3 carbons right here, we can see where they were put onto these 6 carbon molecules there by Rubisco. All of the black carbons used to belong to RUBP. So keep track of those carbons and you can watch them flow through this system. So now one of the first reactions that takes place after we've generated that 6-carbon molecule is to break that 6-carbon molecule in half. So all 3 of these are broken in half. And so then we end up with all of these 3-carbon molecules. So again, keep track of where the red C's are because those are the ones that directly came from carbon dioxide from the air. We use some ATP. We phosphorylate those molecules. Remember, phosphorylating something energizes it and gets it all ramped up and gives it potential energy to do what it's got to do next. So what does it got to do next? Now we got to start adding in those H's. So this is where NADPH delivers the H's. And because we're doing this many turns of the cycle, we're doing 3 turns of the cycle, we're going to need 6 NADPHs. So we need 6 H's here. And of course, remember when we add an H to something, we've reduced it. So that's what's going on in the corner over here. We're showing how we add H's. All right, there's our molecules. And so really what we're going to do here, after we've used our ATP and got our molecules energized so that they can accept their H's, now we're going to take one of them off. So watch this guy right on top. We're taking that one away. That one is going to go on right now to become sugar. Or it's actually called G3P to begin with, glyceraldehyde 3P. It's the precursor to glucose or whatever carbohydrate we want to make at the time, but we usually say glucose to make it easier on ourselves right now. So that one is off of the cycle. That one can leave and can now become sugar, G3P, yeah. So I took one away. I'll show you that again. Oops, it's not cooperating here. So there's my molecules. There I'm taking it away. I'm going to use it to make glucose and whatever else, whatever carbohydrate I need. And then I'm going to reuse ATP. There it is. I'm going to need some more ATP. It takes more ATP for this process than it does the NADPHs. So I'm going to re-energize my leftovers. Where's my leftovers? That's these guys right here. 1, 2, 3, 4, 5 3-carbon molecules right down there. Remember, we're actually doing 3 turns of this to get the molecule of sugar here, G3P. And oh, now hold on a minute. I'm in the way. I got to remove myself out of the way. So after I energize with ATP, now I've regenerated my RU, or after I've used my ATP, I've regenerated my RUBPs. All right, now I'm ready to do that cycle all over again. Wasn't that fun? So all in all, it took 3 turns of this to get one molecule of G3P, which will go on to make glucose or whatever carbohydrate I want. If I'm a plant, I can choose what I want. At the beginning, we took carbon dioxide out of the air. We fixed it onto RUBP. Rubisco was the enzyme that did that, and ended up with a 6-carbon molecule. Then we broke that in half and added a phosphate group from the ATP. So we energized our molecule, which then sets it up so that it accepts the H's off of NADPH. So basically we got to add those H's, and then we make a molecule of G3P. And then anything left over gets made back into RUBP again to keep that cycle going. And again, that is the Calvin cycle, the synthesis part of photosynthesis. This is happening again in the stroma of the chloroplast. G3P actually is a molecule that showed up in glycolysis, if you were wondering. So to G3P and beyond. So we've seen G3P before because that was in glycolysis. It's also in the Calvin cycle. So it's our end product of the Calvin cycle. I know we usually say glucose, but it's actually G3P. It's energy rich because, well, it has carbons and it has hydrogens, oxygen. This is for your run-of-the-mill photosynthesis called C3 photosynthesis. Just kind of your average type of photosynthesis. There's a couple other types that we'll look at, and we can use this G3P to make whatever carbohydrate we want, even turn it into fat if we want. Remember some of the uses of carbohydrates that plants have. They store starch. They need to build a cell wall out of cellulose. They need glucose because they have to make ATP in their mitochondria as well. So this G3P stuff becomes pretty useful. And plants and other producers can make their own. How remarkable is that? And they can even use it in making proteins and DNA and RNA. So Rubisco looks like kind of a big blobby enzyme, is such an important enzyme that it grabs carbon dioxide from the air. Wow, and can then fix it into organic molecules that we can use. More importantly, producers can use, literally making life out of air. Kermit the Frog is green with envy. So let's do a little accounting. So it takes, again, 3 turns of the Calvin cycle to make one G3P. That's why we had to have so many of those little circles with C's in that Calvin cycle diagram. So it takes 3 carbon dioxides to make one G3P. So if we're talking glucose, that means it's going to take 6 turns of the Calvin cycle to make one glucose, if we consider it that way. So that means it takes 6 carbon dioxides to make a glucose. And then in terms of ATP, it takes 18 ATP and 12 H's, or NADPH's, to make one glucose. If there's any ATP left over from the light reactions, well, ATP is also is useful anytime. So we're not worried about leftovers with ATP. So here is our summary with our wonderful diagram. So the light reactions produce ATP and NADPH. They consume water, though, in order to do that. But after consuming water and splitting water, we're left with a wonderful byproduct of oxygen. In the Calvin cycle, the synthesis part of photosynthesis, we consume CO2 out of the air. And that's important for keeping carbon dioxide levels down on Earth. We produce G3P sugar, which can go on to be made into other carbohydrates. And well, we end up with 2 byproducts, ADP and NADP, but that's okay because those can go back to the light reactions in the thylakoids and get regenerated back to ATP and NADPH again to live another day, right? So here's our equation, that it takes water and light energy in the thylakoids of the light reactions to make ATP and NADPH, and then the byproduct, the waste, O2. In the Calvin cycle, the sugar building reactions, we're going to need carbon dioxide and ATP and NADPH, the last 2, which come from the light reactions, and we're going to be able to make glucose, or we could say G3P, and well, then the wastes, ADP and NADP, but that's okay. They can go back to the thylakoids and get recycled again into ATP and NADPH. Putting it all together, the summary diagram, sunlight helps to fuel our energy building reactions in the thylakoids. Water acts as that replacement of the electrons and H's lost from the chlorophyll. So we water our plants to keep all of this going as well. But we generate oxygen, so that's a plus. In the thylakoids, we make NADPH and ATP. That can go then to the Calvin cycle, where we can fix carbon from the air thanks to Rubisco, the enzyme, and then destabilizing those molecules with ATP, phosphorylating them so that they accept the H's that we give to them from NADPH, and then build G3P, sugars. So plants make both energy in the form of ATP and NADPH, and they make sugars. Feeds the world, right? And allows the world to breathe. So there's our summary diagram. Notice that this equation using glucose is kind of a bit simplified. It should actually be G3P right there, but it's easier to say glucose. Plus, glucose is so important in cell respiration, we just like to say glucose. So the great circle of life, how producers can do all this stuff for us. So we should know the answers to these questions. Where does the CO2 come from? The air, right? Where does the CO2 go? Well, it gets fixed into organic molecules and eventually goes into sugar. Where's the H2O come from? Well, that was in the light reactions and watering your plants or from the soil. And the H2O gets split. Where does it go? Well, it gets split and the O goes back to the atmosphere, and the H's from that will eventually go to hopefully the Calvin cycle and eventually go into sugar. Where'd the energy come from? Well, sunlight ultimately, right? But also making ATP during the light reactions. And it's used for the Calvin cycle so that we can phosphorylate our molecules there. Phosphorylating them gives them potential energy to accept the H's that we want to give them, that we gather from the chlorophyll and from the water. What will glucose be used for? Whatever. Food, building stuff. Who knows? I'll eat it. Where did the O2 come from? That comes from water. Where does it go? The atmosphere. What else is involved? I'm sure you can think of some things here. So supporting a biosphere, looking at that on a global scale, photosynthesis is maybe the most important process for the continuation of life on Earth. Each year, photosynthesis captures 121 billion tons of carbon dioxide, whoa, and synthesizes 160 billion tons of carbohydrate. So we're all hungry, we all got to eat, right? So heterotrophs are totally dependent on plants and other producers for their food sources, fuel for raw materials and all kinds of stuff. Not even to mention for breathing, right? Oxygen. So the poetic perspective, all the solid material of every plant was built by sunlight out of thin air, literally out of carbon dioxide from the air. So all the solid material of every animal was built from plant material. So all the plants, cats, dogs, elephants, and people are really particles of air woven together by strands of sunlight. I am now a poet. Very good. So looking at our learning targets here, we've covered a bunch of them. I'd say we've covered 1 through 6. We've still got to talk about, there's some problems with photosynthesis. No process is perfect, right? Evolution doesn't produce perfect organisms or perfection. So there are some problems. One of those is called photorespiration, so that will be a separate set of notes. And another thing is, remember the problem with stomata being open and losing water through transpiration? Well, there's some other adaptations that different types of plants have come up with to kind of help with problems with stomata. So those are called C4 photosynthesis plants and CAM plants. So that'll be a separate set of notes as well. Let me know if you have any questions.